record this. Again, in college, most professors are not going to record their lessons. They're just not. If you miss a class, you missed it, which is freaky, right? Because you're paying $53,000 a year. You got a test next week. You don't have time to miss a class, but you're sick, or you got your mom sick, or you missed it because you slept in, or whatever. Now what? You got to find a way to get those notes from somebody. Again, back to your friends, back to your study group. Study groups have been shown, especially in professional school, study groups have been shown to be strongly correlated with success. All right? Study groups, not friend groups, right? Of course you can fool around and talk. I mean, that's part of being human. But it's about focusing on getting the tasks done that you're asked to do in class. So if you start to form study groups now, you'll see some of the more successful 10th graders, you'll see them in small study groups. They're studying after school. I've seen them. They're going over and reviewing material. Why? Because they want the Josie Edwards Scholarship. They want $500,000. They want to go to medical school, and they know it, right? So you have to find your colleagues that are of the same mindset, of the same kind of focus that you have, right? And what I found in my study groups in college and medical school is that when I'm teaching someone, when I'm explaining something to someone, and I successfully explain it to them, I understand it better. So it's not a bad thing that someone in your group doesn't understand. It is a bad thing if somebody in your group expects you to do all the work. And then that's no good. All right? You can't spend all your energy on other people. You have to spend a lot of it on yourself. But you helping someone else can actually end up helping you. So consider that as you're in a study group. Just because someone's not helping you helping someone else is helping you.
Pardon this interruption. We have contractors here working on the fire alarm system again today. So if you should hear the fire alarm, please disregard it. It is not an actual fire drill. Thank you.
So DNA, deoxyribose nucleic acid. That's what he talked about yesterday. And let's walk through some of the stories he said and kind of make the connections of what he was trying to make with you. There was a time when, let me start with what we know, because they knew something at the time. You know it as well. If you've looked at your handouts, you see that when in your biomolecule handout, you see that you have lipids, right? And the one lipid that we looked at was the triglyceride. I'm just sketching it out. I'm not going to worry about all the hydrogens and oxygens. I just sketching it out here. The triglycerides, some saturated, some unsaturated. Right. Remember, if it's a double bond, that will be on the test. So remember, if there's a double bond, it's unsaturated, right? We covered that, yes? We feel comfortable that you'll be able to tell the difference between saturated and unsaturated fatty acids. This one's unsaturated. This one is saturated. And the word for this is triglyceride. So thank you. Tri means three glyceride bonds, so 3 fatty acids.
So if you see like an equal sign, it's unsaturated?
The equal sign is a double bond, right? So it's sharing 2 pairs of electrons. So it's unsaturated. So if you see a double bond, yes, it's unsaturated. But let's not call it an equal sign because it's two. That's all right. You're right. It looks like an equal sign. But it just means you're sharing two pairs of electrons.
Thank you.
Yes, bless you. So this was the example of lipids, right? And there's some others there. I think if you look, we know that they don't have a monomer. They don't have there's no single unit that you can repeat over and over again to make a larger molecule. We don't, fatty acids in a sense can be put together to make something bigger, but they're not really, there's no real polymer. There's really no polymer. So lipids don't have are not polymers. So the one biomolecule is not polymers. Then we looked at proteins. And proteins have two hydrogens, and they have a central carbon, and then they have this R group, and then they're connected to another nitrogen, right? Well, that's not true. They're connected to a carbon that's then has a double bond, right? So when you have an amino acid, you have this going on. And when you connect it to the next amino acid, this is a monomer. This is a monomer, so this repeats. And this connects another amino acid. So these amino acids are going to become very important. And that's a carboxyl group at the end. So again, you have just to repeat what he had said earlier, that this is your acid part. It's a carboxyl acid. That's one of the functional groups you need to know. So these are both carboxyl groups. I hope you see that. And then you have an amine group here. So together they're an amino acid. Oops. So you have two amino acids when you're putting them together. Let me ask you a question. I think it was in that video that I had you watch for homework and extra credit, right? To watch the video and take notes on it. One of the things that you had to do is see how do you put these together. The way you put them together is you have to, in order to bond these, to make a bond, carbon can only make 4 bonds. As 1, 2, 3, 4. So carbon's already is bonded to everything it can. Nitrogen can make 3 bonds, 1, 2, 3. So nitrogen's bonded to everything it can. How do we bond these two together? You have to get rid of something. That's right. I heard some people say it. You can only create a double bond or triple bond if there's room. There's no room here because, as I said, carbon can only make 4 bonds. How many bonds does carbon have? 1, 2, 3, 4. So that's full. Nitrogen can only make 3. 1, 2, 3. So it's full. So how do we bond these together? We got to get rid of something. So what happens is that in almost every polymer in your body, this leaves in the form of water. And when that leaves, if you watch the notes, you'll see that this is the case. When that leaves, what you're left with is, I'm just going to go ahead and redraw it here, a nitrogen, right? You have a nitrogen, you have 2 hydrogens connected to a carbon, connected to a hydrogen, connected to, again, that R group, connected to a carbon, double bonded oxygen still there, so that's 3. The fourth bond, now the carbon is free to do what? To bond to what? To the other amino acid, to the nitrogen, right? And the nitrogen can bond because it got rid of the hydrogen. This carbon can bond because it got rid of the hydroxide. So now you can form, you still have a hydrogen there. That's a carbon. There's your hydrogen. There's your R group. There's double bond O, and the OH. So now you have just 2 amino acids, but guys, you can keep doing this for very hundreds of amino acids long. You've been doing this for a long time. You can have, I'm gonna go ahead and draw in just circles. I'm gonna draw just 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is just 10 long, but understand that these 10 can, it can be hundreds, okay? In fact, why don't I just make the point by actually just cut hundreds long. It doesn't have to be 10 or 2 or 3. It can be many long. And you take, all you got to do is remember that within each of these is a nitrogen. Oops. Within each of these is a nitrogen, hydrogen, carbon, hydrogen. There's an R group. There's a carbon, double bond, oxygen. But then there's that bond, because you got rid of water, you have that bond between the carbon and the next nitrogen. So when these are all bonded together, oops. These are all bonded together with those electrons, those 2 electrons being shared between carbon and nitrogen all along the protein. So they're linked in a chain. Do you see the chain? Like putting beads together. But the beads are actually amino acids. Now the interesting thing is that these proteins, these R groups, they can equal. This is an equal sign, but instead of using equal, because I just said we don't want to confuse it with a double bond, so let me go ahead and just put an R group can be 20 different groups. There's 20 different ones. There's big ones. There's small ones. There's hydrophobic and hydrophilic and acidic and basic. There's all kinds of different interesting chemistry. This makes them very interesting. They can do a lot. And the key word is do a lot. Very functional. In fact, most of what you consider your body, your hair, your nails, your skin, right? The stuff that you see, most of it is proteins. If it's doing something, it's probably a protein because they're so functional, because they have so many different side groups. Like, you ever see a Swiss Army knife? The proteins are like Swiss Army knives. You just pull open a can opener. You can pull up a screwdriver, get all kinds of things. Even some have little hammers. So you can have all kinds of different things that proteins can do. Why? Because of these 20 different groups. Then we talked about carbohydrates, didn't we? We talked about carbohydrates. And carbohydrates, remember, are basically just C, H, and O in a 1 to 2 to 1 ratio. 1 to 2 to 1 ratio. Every 1 carbon, you get 2 hydrogens, you get 1 oxygen. We talked about that. That's an interesting property of carbohydrate. Mostly carbon and hydrogen and oxygen, that's a carbohydrate. And we drew a bunch of them. We talked about lactose. We talked about disaccharides. We called them saccharides. We talked about lactose. We did a whole little stories and examples of people that are lactose intolerant, correct? We talked about galactose. It's another one of those monomers. The monomers are monosaccharides. saccharides, right, with carbohydrates. They're monosaccharides. And the monomers for proteins are amino acids, right? These are the monomers, the building blocks. You put them together and you get a protein. You put together a monosaccharide and you get polysaccharides. So what are the monosaccharides? The monosaccharides are this hexose thing. There's 6. Let's go ahead and, if you drew it, it would look something like you have an oxygen, you have a carbon, you have another carbon, you have 6 carbons. Oops. It's horribly drawn, forgive me. I'm gonna have to actually erase it. I'm sorry. I hate wasting time, but I just can't. So then each of those has hydroxides, but that would be a glucose molecule. A fructose has the same number of carbons, but it's bonded to the second carbon, so you have this carbon that's sticking out there. And what you end up with is a pentagon, even though it's still 6 carbons. They're both hexoses. If you count them, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. They're both carbon, but this is known as fructose because it forms a 5-ring structure. It's going to act differently, right? Things that are round act differently from things that are square, right? You can't really use a square ring. You pretty much need a round ring. Fructose. I'm just gonna have to use it like so. So that's one. Now there's another monosaccharide becomes very important. Is a 5? These are hexoses. I'm gonna put the word hexose. Remember, ose means sugar. I'm gonna draw now a pentose. What do you think pentose means? This has 6 carbons. What do you think pentoses have? That's right, 5 carbons. There's many different pentoses, but the ones we're interested in look like this. There's an oxygen, there's a carbon, there's a carbon, there's a carbon, there's a carbon, then there's a carbon. And now count them. There's 1, 2, 3, 4, 5. There's obviously an oxygen off of each of these, OH group. Remembering that each carbon needs a hydrogen, needs 4 bonds. Then we know that this has to be happening. So you know that each carbon has to have 4 bonds, so it's mostly carbon and hydrogen. If you count them, how many carbons, if you have 5 carbons, how many oxygens would you expect? 5, because it's 1 to 2 to 1. And how many hydrogens? 10. So let's count. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So 5 oxygens, 5 carbons. That's great. 1, 2, 3. Oh, I missed one. Do you see how I missed one here? Every carbon needs 4 bonds. So 1, 2, 3, 4, 5, 6, 7, 8, 9. I'm missing one. Where is it? This carbon only has 3 bonds. You notice how I caught my mistake there? If every carbon has 4 bonds, then I have to have 10 hydrogens. I have to have 5 oxygens. That's it. So this particular molecule is known as ribose. It's a sugar, so it ends with OSE, ribose. Now, interestingly, if we have the same exact sugar, notice I just copied it. Copied and pasted, that's all I did. Same exact sugar, and I got rid of this oxygen, so that all there is 2 hydrogens. Then it's not ribose, what is it? See, I was wondering if somebody caught it yesterday because he talked about it yesterday. Without an oxygen is called D.
Is it
Say what? Say it. Deoxy. Deoxyribose. So there's ribose, and then there's deoxyribose. The only difference, if we count them, remember how we count these structures? We count them 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. That second carbon, whether there's an oxygen or not, that tells you if it's deoxy or if it's ribose, or is it deoxyribose. It's just that second carbon, whether there's an oxygen or not. That changes the sugar. That makes a big difference, and we'll see in a minute. But those are monosaccharides. So there's deoxyribose. Here's ribose. Oh, I missed this guy. What? What's happening? No, I don't want that. Why did it do that? Anyways. There's ribose, there's deoxyribose, okay? So what's the other group that's left that we have to talk about? We got lipids, protein, nucleic acids. That's right. Now remember, these are polymers. By the way, you can make a polymer of these as well. Take a bunch of these, put them in a long chain, and you got that stuff that you sit on, those wooden stools, that paper you write on. It's called cellulose. Cellulose is a polymer of carbohydrates. That's found in plants. It's a polymer of carbohydrates. I'm not going to go into the details, but look, guys, all you got to do is make a chain of these monomers, which you can change out the monomers and change the quality of the polymer. But so if you used all glucose and you link them up one way, you get this thing called cellulose. If you link them up another way, you get something called starch. You can eat starch. You cannot eat cellulose, right? Nobody's going to sit here and eat paper. I mean, you might have known someone in elementary school who did that, but no, people don't eat paper. At least they won't digest it well, okay? They won't be able to break it down. But if you eat starch, you know starch as potatoes and yams. That you can eat.
Don't starch affect your iron?
Say what?
Don't starch affect your iron?
I'm not sure about that. But I'll get back to you on it. Let's discuss it after class. Let's look at nucleic acids. That's the last group. And nucleic acids, what they are, before we get into what they are in the drawing of it, I'm going to start like he started and tell you what people used to think they are. When people started thinking about heredity, the word is heredity. The word inheritance comes from it. Or is related to it, I should say. Heredity. What do you think heredity means? You're right. In this case, it is the genes, but in general, it means something you get from the people that came before you. Your parents, your grandparents, your great-grandparents, what have you. So you're right. In this case, it would be genes, but back in the day, back in the 50s, they didn't know that. They're like, look, we know something gets passed. They called it the transforming, and you know what transforming is from the movie Transformers, right? Something that changes. The transforming principle. And they don't mean And like you said in the video, they don't mean like the way we normally use principle. It's used in an antiquated way, in a way that we don't use it anymore. But they mean the stuff of inheritance. What is the thing that you inherit? Like, you know, you got your mom's eyes, your mom's nose, you got your dad's height, you have your dad's earlobes, you know, whatever. You know you got that. But what is it? How did he give you that? When you were in the womb, did your mom go in there with a little scalpel and a hammer and start pounding your nose in a certain shape?
It's in the genes, so basically, you have 50.
Right. We know that today because we taught it to you. You're right. Everything you're saying is correct. What I'm saying and what he was trying to say yesterday is back in the day, they didn't know. They were like, look, it has to be one of these 4 biomolecules. When we take a person, we grind them up in a blender, right? And we separate the pieces, we find four different kinds of molecules. We find carbohydrates, we find lipids, we find proteins, and we find nucleic acids. Which of these 4 is the stuff that gets passed on from parent to child? We didn't know at the time. They were thinking that most people thought DNA was too boring. It can't do a lot of stuff. It's not very useful. It's not like a protein. Protein can be an enzyme. It can be like your fingernails hard. It can be soft like your hair. It can be soft like your skin, or it can be hard like your fingernails, or a horn, right? Like a antler. Proteins can be all kinds of things because there's 20 different kinds that you can put together in all those different ways. DNA is boring. There's only 4 kinds. I'm about to draw it for you, but there's only 4 kinds, he says. 4 kinds of DNA. Yes. So why it doesn't make sense that DNA would be the thing you inherit from your parents that if you want to think about it this way, if you're going to write a book, someone volunteer their name for the sake of the video. Go ahead. So James. If we're going to make James, we're going to build James. We're going to start with a single cell. We need instructions, right? We need instructions to when we're building. Like, if you're going to build a house, don't you need instructions? Don't you need need a blueprint? You need something to tell the plumber, look, put the toilet here, right? You don't want the toilet in the bedroom. That makes no sense. You need to have bedrooms here. I need you need a plan. Well, James had a plan. He got it from his parents. That plan was written. It's written in a language. It's a language. It's written, today we know it's written in DNA, correct? But back then, if you're going to think about writing a book as complicated as James, right? James is very complicated. He has 2 eyes. Each of those eyes are connected to a brain. They have all kinds of molecules that collect light. I mean, you created 2 eyes. They're so complicated. They can see light. And a brain that's so complicated, they can take that light and interpret it into a picture of what's going on. You have ears with all little tiny bones that pick up sound, and you translate that sound into something that makes sense. That's insane. That's insanely complicated. You take food like a steak or an apple or salad, and you break it down to the most smallest possible molecules. You absorb it into your blood, and then you make more of yourself. That's complicated. It's incredible what you're able to do. You have all kinds of organs. You can breathe air. You can run. You can swim. You can think. You can make tools. How are we going to make James? You need a complicated book. Don't you think the book is going to be pretty big? Let me tell you something. It's billions of letters long. If this were James's book, it would, and we stacked them one on top of the other, it would reach the moon. That's how big the information is that makes James who he is. And that's true for all of you. Because you're so complicated. So we need to know, knowing that, would you choose, if you're an intelligent human being back in the 40s, would you choose DNA only has 4 letters, or would you choose something like proteins that has 20 different letters? Proteins. Why? I mean, we know it's DNA today. You're correct. But we would have guessed, and every intelligent person guessed proteins because they're so complicated. But it turns out, if you think about it really deeply, you have to be reflective. It turns out it is DNA, and the answer is kind of simple. You want that book to stay stable. You don't want, when you're making the copies, because you went from one copy of a book that goes all the way to the moon, and then you created 1 trillion copies, because you have about 1 trillion cells in you. And you created 1 trillion copies of those. And you're constantly making cells, so you're constantly making copies. The one thing you don't want is there to be a mistake in this message, right? You don't want all of a sudden James starts growing another head off his shoulder, right? That'd be kind of creepy and weird and wouldn't function very well. You wouldn't want James's anus to be out of his throat, right? Or his nose on his butt. That would not make sense, correct? It wouldn't work. You want to make sure that the message stays stable. You don't want it reacting. You don't want it changing like crazy. If you made it out of a protein, it could do a lot of stuff, but you would then you take risks that you're going to change change it too much, correct? Like when you make your rings, why do we use gold? Gold's pretty boring. Gold is incredibly boring. Why do we use it for jewelry?
Because it's not that easy to change or break.
That's right. It's not that easy to change. I can stick this hand. I stick it in bleach all the time because I'm constantly washing things. I don't have to worry about this changing. If I made this out of tin, nickel, copper, even silver, it would change, wouldn't it? Yeah. If it was copper, it would turn green. And it's interesting, isn't that interesting? It's much more interesting. It does a lot more. Copper does a lot more than gold. Gold is boring. It's not going to change. But it's going to last longer. And that's why DNA is the code of life, because you want it to last long for your whole life. In fact, you want it not just to last your life, you want to pass it on. And you want it to stay the same from generation to generation. You don't want humans to have 2 arms and 2 legs in your generation, and then the next generation have 3 arms and 3 legs, and the next generation you got 6 arms and 6 legs. That wouldn't be any good, would it? Like, that doesn't make sense. So we need to have something that's stable, like gold. Nice, stable, secure, that lasts a long time. Yeah.
With the formities that do have another head on them, how does their DNA work? How does that happen?
That's a really good question. People that are born with multiple heads are what we call Siamese. Well, they're not called that, but people call them Siamese twins. They are people that were two people growing together in a womb. They got too close, and the cells started sharing space and sharing messages. They were messaging each other, and they became, in a sense, one connected, strongly physically connected. They're still separate human beings. Their brains are separate. They think differently. They feel differently. But they might share a liver. They might share a hip. The lucky ones don't share a heart and don't share a spine. Because if you share a heart or a spine, then you can't separate. But if you share just a kidney and a liver, then and you have enough bone, then you could even get rid of one person could lose even a piece of the hip. You can replace it with an artificial hip. But if you share an irreplaceable organ, they're together for the rest of their lives, which isn't a horrible thing. There have been people that have been congenital. We call them congenital twins that are born that live their whole lives together. But imagine though the struggles that they would have to have. Like, it's something I can't even think about doing. But yeah, that does happen. That's not genetic, right? That's not a gene thing. That's a growing up thing. That's an environmental impact.
Well, like, what happens, like, 'cause I've seen, like, how these two people, their faces are connected, but they have different bodies.
Yeah, when it's the same thing. They were twins. They got too close in the womb, and they started growing into each other. So what we can do today with our technology today is separate them. And we can reconstruct the jaw. We can do facial reconstruction. We can take tissue from the leg, for instance, and move it up to the face. You can do stuff like that and rebuild the things that were taken away, right? In the 50s, 40s, and 30s, that was not possible. So if you were born connected to the face, you stayed connected the rest of your lives. In most ancient cultures, they would be called, they would be holy. In a lot of cultures, they'd be holy. Some, they would have just killed them at birth. The Spartans would have probably thrown them on rocks. But they might have been considered holy people and people connected to God or whatever, just as many schizophrenics were considered holy as connected to God in some way. Yeah.
If that's what happens, like when you're born, do disabilities have something to do with genes? Like
They can. That is a really good question. It's something we need to think about. It's something kind of a contextual thing, because a lot of people have this misconception that every disease that babies have is something they inherited. That's not true. There's different reasons people have sicknesses. For instance, you could be born with something called cerebral palsy. It can impact the way you think. It can impact your body. Some people with cerebral palsy are impacted physically. Some are impacted mentally. My aunt was born with cerebral palsy. She was a twin, and she was born with cerebral palsy. She had problems with her thinking. She was physically fine, but her thinking was off. All right? That is not genetic. That is lack of oxygen in the womb. So if something happens and the umbilical cord gets wrapped around the neck, or oxygen somehow is during birth is the oxygen to the brain is impacted, then that person can be born with damage to the tissue. That is not genetic, right? That's another one of those environmental situations, just like the twinning that we talked about a minute ago. Vertigo, I'm not sure. I'm gonna guess. It's just a complete guess. Is genetic in some way. Now, is there environmental? A lot of diseases are both genetic and environmental. For instance, high blood pressure, heart disease. Heart disease. My father had heart disease, but he smoked, right? And smoking, we know, causes heart disease. But then he was also, but there's people that smoke their whole lives that don't get heart disease. Well, what's the difference? Well, their genes, for some group, makes them more susceptible than others. So it's complicated because it's not all one-to-one. Sometimes one thing changes and it makes you more, what's an example? Okay, so skin cancer is caused by the sun touching your face too much, the UV radiation penetrating your DNA and changing it. However, people that are born with darker skin have protection against that, natural protection. People that don't have that darker skin don't have that protection. But you both can get skin cancer. Does that make sense? So one is more susceptible than another because of your genes. And that's true of a lot of diseases. You don't necessarily get the disease from your genes, but your genes can make you vulnerable to those diseases.
Wait, is diabetes genetic?
Type 2, we know type 1 is. Type 2 it runs in the family as well. There's a genetic component, but there's an environmental component as well. Eating too much sugar repeatedly, being overweight, causing your body to be inflamed, having inflammation, not getting exercise, not taking care of you for a long time, can cause your genes, right? Because of your genes, cause you to be diabetic. Other people do exactly the same behavior and they never get diabetes. or at least they get it later in life. All right. So that is something that you need to consider, okay? But the transforming principle they thought was proteins, but it turns out it's DNA. Let's take a look at what it is. Do we have time? Well, I can draw this, the thing he drew, and then we're done. And by the way, great job today. Great job today. So oxygen is connected to carbon. We're going to draw a deoxyribose because we're going to draw DNA. So now this is a deoxyribose because it has hydrogens here. I'm not going to draw all the hydrogens, okay? I'm just going to draw this because we're running out of time. Up here, of course, you have an OH group and an H, two H's sticking out here. Down here, you have also an OH and an H. Now, if we don't, DNA's monomer is not ribose, but it's something called a nucleotide. Now, when we take, and again, you have this drawing in your packet. If we take this carbon and we connect it to a phosphate, that's a phosphorus, connected to an oxygen, connected to 4 oxygens. That's a phosphate. To make it simple, this PO4, I'm going to draw it from now on. You're going to see me draw it like this. Is that okay with everybody? Because it's too many. You saw him draw ATP with all those phosphates. You all freaked out. You're like, what is that? How do you draw it so fast? What is that? It's ATP is what it was. So I guess we'll finish this tomorrow, but just know that right off here is the nitrogenous base. And that's called a nucleotide.